Hi everyone. We're going to be doing 9.16 to 9.18, and most of this will be review from a lot of the concepts we've learned so far. Okay, so there's something called the chromosome theory of inheritance, which states that genes will occupy a very specific loci or location on the chromosome, and that chromosomes will go through segregation and independent assortment during meiosis. These are all things that we've already talked about, but this theory of inheritance is pretty much just saying that chromosomes actually do follow all these kinds of behaviors. So because the chromosomes have certain characteristics, like the fact that genes are on very specific locations on the chromosome, that allows chromosomes to be able to kind of do crossing over and switch their alleles, so that it increases genetic diversity. Something else about chromosomes that we've talked about is that during meiosis, the parent cell only donates half of its genetic material to each gamete. And then remember, those gametes recombine to form the full offspring. And so that is an aspect of chromosomes that leads to the patterns of inheritance that we observe in real life. And the other thing that affects the inheritance patterns we see is the fact that chromosomes can randomly align or go through random independent assortment during metaphase 1 of meiosis. And so that just means that the gametes have almost infinite possibility of having different combinations of chromosomes from the mom and from the dad. And so that's also something that affects the patterns of inheritance that we see. Oh, I couldn't think of a good picture, so this is just a cute picture of a dog. Okay, something different than what we've talked about that actually opposes independent assortment is the concept of linked genes. Linked genes are when genes are located so close together on the chromosome that they have a tendency to be inherited together. So if you can imagine a gene, this is an example. If you can imagine a chromosome, you see how these genes here are so close together that when there's crossing over, they tend to be crossed over together, the P and the L. And so what you'll notice when there's linked genes is that instead of seeing the 9:3 to 3:1 ratio for the offspring in terms of its phenotype, so you'll notice here that most of the offspring will exhibit just the characteristics of its parents, and you don't see recombination where there's a big P and a little L. You don't really see that very often, and you also don't see the opposite, little P and big L, because the recombination isn't really happening. So as we said, they don't follow the law of independent assortment, which means that these genes tend to be inherited together. You're more likely to get offspring that look like the parents, and that this skews the ratio we had talked about earlier. This 9:3 to 3:1 ratio is assuming that there's independent assortment and that there's complete dominance in the traits. So an example of linked genes, red hair and freckles tend to be linked together, which is why you'll see a lot of people who, when they have this kind of orangey red hair, they tend to also have freckles. Okay, so just to review some of the concepts we talked about, crossing over, which occurs during prophase 1 to kind of metaphase 1 of meiosis, crossing over is what leads to genetic recombination. And so you'll have some cases where genes will not recombine, and then they won't do any crossing over, they won't recombine in certain sections. And so you'll have parts of the chromosome that look exactly like the parent cell. So in this case, this right here and this right here replicate just kind of what you see in the parent cells. But these two combinations where you have a big A and a little b are the results of crossing over, and a little a and big B. So there was someone who did an experiment with flies that kind of showed how crossing over and linked genes kind of look like in real life. This person did a test cross between a heterozygous fly and a recessive fly. So this fly right here is heterozygous, It exhibits the dominant traits for gray body and long wings, but it's dominant in the heterozygous form. And it was crossed by a homozygous recessive, so it's black and short wings. And so if the genes were not linked, so the genes for the color of the body and the length of the wing were not linked, you would expect a 1-to-1 to 1-to-1 ratio. So that just means that all of these kinds of offspring would have equal probability of being seen. Gray and long wings, black and short wings, gray and short wings, and then black and long wings. But this is not what this person found. This person actually found that there was unequal ratios, and then the offspring really favored the phenotypes that looked exactly like the parents. So you notice that this one right here, the gray and long wings, and then the black with the short wings, were much, much more likely to be seen than the other two. So this actually shows that the genes are most likely linked. You're most likely to inherit those two genes for the color of the body and the length of the wing together. But there is still some recombination that is possible. So it's not impossible to do crossing over. It's just less likely because they're close together. And so this recombination frequency, which is just the frequency of these phenotypes or these alleles, these genotypes that are different from the parents, is about 17% for this experiment. So this is all the new information for the reading for today. It's mostly just a review of concepts that we've already talked about. Thanks for watching. Goodbye.